Okay, welcome to the deep dive. Today, we're pulling back the curtain on something truly fundamental in modern medicine. The incredible process that takes, raw genetic data from a patient's sample and transforms it into clinically useful information.
It really is a remarkable journey for that data. You start with billions of these individual DNA letters, and somehow you end up with a clear picture that can actually guide diagnosis and treatment.
And what's really striking is how much of this is automated, right? Like a complex recipe.
Exactly, a sophisticated digital workflow. Often it's orchestrated by a master recipe encoded in a script, like the bash script we're looking at.
Right, so this script is the engine, basically, turning all those A's, T's, C's, and G into meaningful insights, crucial for things like inborn errors of metabolism, for example.
Absolutely.
So our mission today is to unpack the purpose behind each step in this automated pipeline, using the script you shared as our guide. Let's dive in.
Sounds good. So to understand a patient's unique genetic code, you first need context. You absolutely need maps and reference points.
Right, like you can't navigate without a map.
Precisely. Imagine trying to understand, say, a unique architectural blueprint without knowing the standard building codes or even having a map of the area. It wouldn't work.
Makes sense.
So phase one is all about setting the stage. It's about gathering these essential reference libraries.
And the absolute bedrock, I guess, is the human reference genome, HG3038 you mentioned.
That's it. That's the standard agreed-upon genetic blueprint of a, notionally healthy human. Think of it as the master map. Every piece of patient data we analyze gets compared against this map to spot the differences.
Okay, so that's the map. But you said not all differences matter.
Exactly right. Many differences are just common, harmless variations found across populations. So alongside that master map, we need these huge catalogs of known variations.
Like dictionaries of variations.
Kind of, yeah. Databases like DBSMP, 1,000 genomes, they list genetic changes already seen in thousands, even millions of people. These are vital for helping us quickly sort of separate a common, likely benign change from something rare that might actually be linked to a disease.
Okay, map and reference books of common variations. But what if you're not looking at the entire genome? Like for an exome sequence.
Great point. Yeah. For targeted tests like exome sequencing, where you're mainly interested in the protein coding genes, you need specific files. These files basically define exactly which regions of the genome you care about.
Like highlighting specific neighborhoods on the map?
That's a perfect analogy. It focuses the analysis only on the parts most likely to be relevant, which saves a ton of time and computing power.
And then finally, these annotation databases. What do they do?
Yes. Think of these as the specialized encyclopedias. So the reference genome and variant catalogs help us find variants and see how common they are. These annotation databases, they come in later, after we found potential variants.
Okay.
They add layers of biological and clinical meaning. They tell us things like, okay, which gene is affected? What might this change do to the protein? Have this specific variant ever been seen before in patients with genetic diseases? That kind of crucial context.
So phase one is all about assembling the toolkit, the library you need for the analysis.
Exactly. Building that essential library of resources.
Got it. Okay, library stocked. Now we get the raw DNA sequence data, fresh off the sequencing machine. It's probably not perfect, right? You can't just compare that directly to the nice, clean reference map.
Oh, absolutely not. No way. Just like any physical sample needs prep and quality control in a wet lab, this raw digital data needs serious cleaning and validation. That brings us to phase two, preparing the patient data quality control and cleaning.
Right. Because, garbage in, garbage out.
That's the mantra. So the very first step is a quality check, usually with a tool like FastQC. It looks at the raw sequence reads and assesses their quality, looking for any red flags, technical artifacts, that sort of thing, maybe from the sequencing run itself.
And if it finds issues, or just like messy bits at the ends of the reads.
That's where trimming and filtering come in. This step is absolutely crucial. You often get lower quality bases at the ends of reads, and sometimes you have leftover bits of technical sequences, adapters that were added during sample prep.
Okay.
Those need to go. Keeping them would really mess up the next stage, the mapping. So tools like TrimGlore are used to snip off those low-quality ends and remove adapter sequences. Reads that are too short after trimming might get filtered out too.
And then, logically, you'd check the quality again after you've cleaned it.
You got it.
Yeah.
A post-trim quality check, often running fast QC again, just confirms the cleaning worked and the data quality is looking good.
Makes sense.
And because you might run QC multiple times on multiple files, there are tools like MultiQC that are really handy. They gather up all the results from the different QC steps and tools and put them into one single summary report.
Gives you the big picture.
Exactly. A quick, high-level view of data integrity. So this whole phase, it's all about refining that raw data, making absolutely sure it's trustworthy before we attempt the really deep analysis.
Okay, makes total sense. Data's cleaned, it's trustworthy. Now comes, I guess, the really big computational step. Figuring out where each of those millions of cleaned up patient DNA fragments actually belongs on our reference map. This is phase 3, mapping the genome alignment and post-alignment processing.
This is where the puzzle pieces really start coming together. Millions, sometimes billions, of these short DNA fragments from the patient are aligned, meaning they're precisely mapped back to their correct location on that human reference genome, HG38.
So finding the address for every single piece.
Pretty much. Tools like BWA-MEM are workhorses for this. The main output is what's called a BAM file. It's a highly organized, standard digital format that represents the patient sequence genome, showing where every single read lined up on the map.
Like laying out all your jigsaw puzzle pieces correctly on the picture on the box lid?
That's a great way to think about it. But even once they're aligned, there's still a bit more processing needed for top accuracy.
Oh. Like what?
Well, one really important step is identifying and marking duplicate reads. During the lab preparation process, purely by chance, some DNA fragments get copied multiple times before sequencing.
Okay. PCR duplicates.
Exactly, often PCR duplicates. We need to find these because if you have 10 identical copies of a read showing a certain variant, you don't want to count that as 10 independent pieces of evidence. That would artificially inflate your confidence in that variant. So tools like Picard mark duplicates flag these.
So you're making sure each unique original DNA molecule is basically counted only once. Smart.
Yes. And then there's another refinement called base quality recalibration, often using JTK tools. This is pretty sophisticated.
Okay.
See, every time the sequencer calls a base an ATC widget, it assigns a quality score, like a confidence level. But these scores aren't perfect. They can have known systematic biases based on things like the machine cycle or the sequence context around the base.
Right.
So this recalibration step uses known patterns of sequencing errors and also databases of common known variations to adjust those initial quality scores for each base. It makes them more accurate.
Wow. It's like fine-tuning the confidence meter for every single letter based on known issues.
That's exactly what it is. And it makes a huge difference later when you're trying to decide if a small difference you see is real or just a sequencing error. It dramatically improves the accuracy of variant calling.
I can see why.
And then typically there's a final quality check, sometimes using GATE's analyzed covariates, just to confirm that the recalibration step actually worked as expected, that the quality scores look better, and the data is truly primed for the next big step, finding the actual variations.
Okay, so phase 3 gets everything lined up perfectly and polishes the data quality to a high shine.
You could say that. It ensures every piece of the puzzle is in its place, and the data is highly trustworthy.
Right, so data cleaned, mapped, refined. Now for the main event, right, the actual hunt for the differences between this patient's DNA and that standard reference genome. Phase 4. Discovering genetic variations, variant calling.
This is it. This is where the software actively scans that mapped, recalibrated patient data, the BAM file, to pinpoint all the specific genetic differences. The key tool here is often something like DHTX haplotype caller.
Haplotype caller. Okay, and what's it looking for exactly?
It scans along the genome, looking at the aligned reads covering each position, and tries to identify anywhere the patient's sequence consistently differs from the reference.
And the main types of differences are?
Well, the most common are single nucleotide polymorphisms, SNPs. That's just a change in a single DNA letter, like an A changing to a G at a specific spot.
Okay.
And the other main type is indels, small insertions or deletions of one or more DNA letters. Haplotype caller is designed to detect both SNPs and indels.
And the output of this step, what does it look like?
It generates a file, typically first a GVCF, then processed into what's called a VCF file variant call format. This VCF file is essentially a detailed list of every potential genetic variation identified in the patient's sample compared to the reference.
So this VCF file is the raw list of potential differences. Every single spot where the patient might deviate from the standard blueprint.
Precisely. It's comprehensive, but it's still considered a raw list at this stage. It contains all the candidate variants the software found based purely on the sequence data.
Which means it probably contains mistakes or things that aren't clinically relevant.
Exactly. That raw VCF list invariably contains a lot of noise. There will be some false positives, positions flagged as variants due to subtle sequencing or alignment errors, and also lots of common benign variants that aren't related to the patient's condition,
Okay.
which is why Phase 5 is absolutely critical. Filtering variants, removing the noise. The whole goal here is to take that raw, noisy list and filter it down. Try to keep only the high-confidence, potentially meaningful changes.
Makes sense. You need to separate the signal, the real variance from all that background noise. How do you start doing that?
Well, first, there might be a normalization step, using tools like BCF Tools Norm. This just ensures that variants, especially complex ones like indels, are represented in a standard, consistent way. It makes comparisons easier later on.
Okay. Standardization.
Then sometimes, for filtering purposes, variants are temporarily split into two groups, SNPs and indels. They often have slightly different air profiles, so applying filters might be done separately.
Ah, okay.
But the core of this phase is applying stringent quality filters, often called hard filtering, using tools like GATK's variant filtration. This is where you apply a set of predefined rules or thresholds based on various quality metrics associated with each variant call.
And tricks. Like what sort of things?
Things like QD, which is quality by depth. How good is the call relative to how many reads cover it? QAL, the overall quality score assigned by the variant caller. SOR, a measure of strand bias. FS, another measure of strand bias. Fisher strand. MQ mapping quality of the reads supporting the variant. DP, the total read depth at the site. And rank-sum tests like MQ-rank-sum and ReadPos-rank-sum that look for biases in where the variant appears within the reads.
Wow, okay. So it's like a really strict checklist for each potential variant. Does it have enough reads supporting it? Is the quality score high enough? Is there weird bias in which DNA strand shows the variant? That kind of thing.
Exactly that. Think of them as quality control gates. Each potential variant has to pass these tests. If a variant fails one or more of these filters, maybe its quality score is too low, or it shows significant strand bias, suggesting it might be an artifact that gets flagged as low quality or filtered out.
So this step dramatically cuts down the number of variants you have to consider.
Hugely. It aims to remove the vast majority of false positives. After filtering, the high-quality SMPs and indels might be merged back together into one file.
Okay.
And then there's often one final selection step. Using those target region files we talked about way back in Phase 1, the ones defining the exome, for instance, we select only the high-quality variants that actually fall within those specific regions of interest.
Ah, so you filter for quality, then you filter for location, making sure you're only looking at variants in the genes you intended to analyze.
Precisely. So by the end of Phase 5, you should have a much smaller, much cleaner list of high-confidence genetic variations located within your target regions, accurate and relevant.
That's a huge amount of refinement. Okay, we've journeyed from billions of raw reads down to this refined, high-confidence list of variants in the right places. But just having the list, a C changed to a T at position X, that doesn't tell you what it means clinically, right?
Not at all. Identifying a variation is just one part. Understanding its potential significance is the next crucial step. And that's Phase 6, interpreting variance annotation.
Annotation, giving meaning to the variations.
Exactly. This is where we translate that genetic change into biological and clinical context. It's like consulting a massive interconnected library of genetic knowledge dictionaries, encyclopedias, clinical reports, all at once. Tools like Anavar are workhorses here.
Okay, Anavar, what does it do with our filtered variant list?
It takes each high-quality variant from our list and enriches it by querying a whole slew of public databases simultaneously.
Yeah.
The script we looked at listed a bunch: RefGene, Nomad, 100,000 genomes, ClinVar, Revel, DBNSFP, tons of them.
Wow. And what kind of information is it pulling from all those sources for each variant?
Really critical context. For starters, gene location. Which gene does this variant fall in? Is it in an exon, protein coding part, an intron, or somewhere else? Then protein impact. What does this change potentially do to the protein sequence? Is it a silent change? Doesn't alter the amino acid.
Yeah.
A missense change swaps one amino acid for another. A nonsense change creates a premature stop signal. Or maybe it affects splicing.
Okay, so location and predicted effect on the protein. What else?
Hugely important. Population frequency. How common or rare is this specific variant in different populations globally? Databases like NomandD provide frequencies from hundreds of thousands of people. A variant seen in, say, 10% of people is almost certainly benign. A variant never seen before or only in a handful of people, that's potentially more concerning.
Rarity is a big clue.
Definitely. Then you have computational predictions. Various algorithms, using information about protein structure, evolutionary conservation, and the type of amino acid change, predict whether a missense variant is likely to be damaging to the protein's function or likely benign. Tools like SIFT, Polyphony, CADE, REVEL provide these scores.
So educated guesses from computer models.
Educated guesses, yes, based on lots of data. Okay. And perhaps most critically, clinical significance from databases like ClinVar. Has this exact variant been reported before in association with the disease? Has it been classified by experts as pathogenic, likely pathogenic, benign, likely benign, or very commonly a variant of uncertain significance, VUS?
So connecting it directly to known clinical findings if they exist.
Yes, exactly. Anavar integrates all this information, location, function prediction, population frequency, computational scores, clinical reports, and adds it directly to our variant list. So each line in the output file goes from just being HHR1, position 1, 2, 3, 45, CT, to having columns detailing all this rich contextual information.
That transforms the data completely.
It really does. And after that massive automated annotation, there's often one more step. Custom filtering. This might be a separate script, maybe in Python, that applies lab-specific or case-specific filters.
Tailored to the patient.
Yeah. For example, it might prioritize looking only at very rare variants predicted to be damaging that fall within a list of genes known to be associated with the patient's specific symptoms or family history. This helps focus the final review on the most likely candidates.
So annotation adds the meaning, and custom filtering sharpens the focus right at the end.
Precisely. This whole phase is about bridging that gap from raw genetic code to clinically interpretable information.
Wow. Okay, that is a truly complex, multi-step journey we've followed. Billions of reads down to a focused, annotated list of potentially significant variants. So what happens after all that computational heavy lifting finally finishes? What are the final outputs?
Well, the whole pipeline really culminates in the creation of that actionable genetic report, or at least the data that forms its core. But before the final files are stored, there might be a couple more quick analyses.
Like what?
Sometimes additional scripts calculate detailed sequencing coverage statistics across specific genes or regions of interest. This helps confirm you actually got enough data in the key areas to be confident about both finding variants and not finding variants, basically assessing the completeness of the analysis in critical spots. They might also run some final genotype refinement steps.
Just adding those last layers of quality assurance.
Exactly. Then comes a really practical but important step: final organization. All the key output files, the recalibrated BAM file, the aligned reads, maybe the GVCF, and most critically, that final annotated VCF file with the filtered, interpreted variants need to be moved into designated permanent storage folders.
Got to keep things tidy and findable.
Absolutely. For clinical use and long-term archiving, the script often uses commands like MV to move files, and sometimes LNN-S to create symbolic links, which are like shortcuts, so files can appear in multiple places without being duplicated.
Smart. Save space.
And then finally, there's usually a cleanup step. All those intermediate files generated during the many steps, temporary alignments, unfiltered variant lists, QC reports that have been summarized, they can take up a lot of disk space. So commands like armm-rf are used carefully to remove these temporary files once the final outputs are secure.
Housekeeping.
Essential housekeeping. And this entire robust workflow, automated by that script, it might run hundreds of complex commands sequentially. It also often leverages parallel processing, analyzing multiple samples at once, or using multiple computer cores threads for single intensive steps to dramatically speed up the whole process from days or weeks down to hours.
So this automation is just fundamental to getting these results back in a clinically useful time frame.
Absolutely fundamental. It ensures efficiency, it ensures consistency, the same steps are run the same way every time, and it ensures reproducibility. It's what takes the incredible power of genomic sequencing and translates it into information that can actually guide critical clinical decisions in this era of precision medicine.
It's just remarkable when you think about that whole transformation from, these invisible bits of data in a patient sample all the way through this complex digital refinery to a filtered, annotated list that a doctor or genetic counselor can actually use.
It truly is. The automation is key to handling the sheer scale and complexity. Without it, analyzing this data would be practically impossible on a large scale.
Which, leads to a really interesting thought to end on. This pipeline, as we've discussed, is incredibly sophisticated. It pulls from vast databases, applies complex algorithms and filters. It does so much work. But the output, even after all that, is still often a list of variants, including perhaps some variants of uncertain significance or maybe even novel variants never seen before.
That definitely happens.
So, given the sheer volume of information processed and the potential ambiguities remaining in that final output, what new challenges do you think emerge right there at the interpretation stage? What's the indispensable role that human expertise, the clinical geneticist, the counselor, considering the actual patient plays after the script has done all its automated work? What happens next?